Hello, everyone. Today is the last lecture from the textbook. Actually, only about 1/3 of the content is from the textbook, and the rest are recent scientific development related to this topic. So when you go over this lecture and prepare for exam 3, please follow the slides and the talk. In this lecture, we will talk about treatment for genetic disorders. Many genetic disorders result from gene changes that are present in essentially every single cell in the body. So as a result, these disorders often affect many body systems, and most of them cannot be cured. Okay. However, approaches may be available to manage some of the associated signs and symptoms. In some cases, enzyme replacement therapy can help compensate for the enzyme shortage. These treatments are used to manage existing signs and symptoms and may help prevent future complications. One good example is the Pompe disease we learned last time. The drug Myozyme, a genetically engineered protein, is given intravenously and it acts like the naturally occurring acid alpha glucidase enzyme. Most treatment strategies for genetic disorders do not alter the underlying genetic mutations. However, a few disorders have been treated with gene therapy. Okay, gene therapy is the transfer of normal clonal genes into individuals with genetic diseases to replace the defective genes. Theoretically saying, gene therapy could bring a cure to a genetic disease. This slide shows how gene therapy is done to treat patients. The most widely used method uses a virus as a vector to insert a normal copy of gene into certain genes of a patient who has a genetic disorder. In the case shown here on this slide, the white blood cells are used. After the engineered virus delivers normal gene to the white blood cells, the normal gene becomes active and the cells are then injected into the affected individual, then curing the genetic disorder. Because white blood cells die after a few months, the procedure has to be repeated regularly. In the future, it is hoped that safe transferring a normal gene into the stem cells in the bone marrow will make gene therapy a one-time procedure. But then can you think about some differences between gene therapy and the stem cell therapy we talked before? Whereas gene therapy involves the transfer of genetic material into the appropriate cells, stem cell therapy is a direct transfer of cells to the patient. In other words, gene therapy involves the transfer of genetic material using a carrier or vector to integrate the gene into the appropriate cells of the body. But stem cell therapy only involves the transfer of cells with the relevant function into the patient, and there's no genetic engineering ever been done. Okay, also, can you think about any other technology that we learned that can be used for gene therapy? Yes, CRISPR-Cas9, the genome editing technology. This table shows the successes and failures on the history of gene therapy. Between 1989 and December 2018, over 2,900 clinical trials were conducted, with more than half of them in phase 1, suggesting this field was not moving very fast. The reason includes 3 patients' deaths in gene therapy trials, putting this the whole field under close inspection. 2 incidents were listed in the table by the blue arrows. It is always important to remember that although deaths are rare, but they can still occur, and it's very possible that certain types of gene therapy can cause certain cancers. This slide shows an overview of global gene therapy clinical trials in our textbook. You can tell that there are more clinical trials for cancer therapy than any other classes of diseases. So the N labeled here means the number of clinical trials. So you can see the cancer trials is more than any other trials added up together. Let me give you a specific example of how gene therapy is done. This slide shows an exon shuffling in gene therapy for Duchenne muscular dystrophy. I will just briefly go over this, as this disease will be discussed in detail in our group project. Okay, in this genetic disorder, a deletion mutation in the exon 52 of the dystrophin gene will occur. So in B, this mutation causes a shift in the codon reading frame in the exons, causing translation to end at exon 51 instead of the exon 79 in the normal situation. So this results in the production of a non-functional fragment of the dystrophin molecule. So from exon 53 to the end, there's not going to be translated, okay? And then the purpose of the gene therapy is to restore the reading frame of this mRNA. So scientists use an exon-specific antisense oligonucleotide that specifically bind to the exon 53, blocking the processing signals for this exon. Okay, so the antisense oligo is a strand of a nucleic acid, either DNA or RNA, or even a chemical analog. So they can bind it to the messenger RNA, the mRNA, and inactivate the mRNA. So as a result, the splicing factors splice out the exon 53 and links the 51 exon to the exon 54 together, restoring the reading frame. Okay, so the resulting dystrophin molecule is shorter, but it is partially functional. At present, gene therapy uses somatic cells as targets for transferred genes. This form of gene therapy is called a somatic gene therapy. Okay, genes are transferred into somatic cells of the body. There are also two other forms of therapy that are controversial, mainly because the ethical issues surrounding them have not been solved. Okay, one of these is germline gene therapy, in which cells that produce eggs and sperms are targets for gene transfer. After germline therapy, the transfer gene would be presented in all the cells of the individual produced from the genetically altered gamete, right? As a result, these individuals' future generations will all be affected by this gene transfer without their consent. We will talk about a related case next. Okay, the other form is enhancement gene therapy, in which gene transfer may be applied for enhanced athletic ability or intelligence. Gene doping is an example, and we will talk about it in a minute as well. Before I introduce the case of germline gene therapy, I would like to give you some background information, okay? So remember we talk about HIV, and this virus targets CD4 positive T cells because it surface glycoprotein GP120 that will recognize the CD4 molecule on T cells. But in order for HIV virus to successfully enter the host cells, it needs another co-receptor on T cells. So its name is CCR5, okay? Interestingly, there is a naturally existing deletion form of CCR5 named CCR5 delta 32. So this deletion form of CCR5 can prevent the entry of the HIV virus. Therefore, even HIV can enter these people's bodies. They cannot infect their CD4 positive T cells. In the European population, about 1% of the people has this mutation. So in 2018, there was a big news about this person. His name is Jian Kui He. He actually obtained his PhD from Rice University and had a postdoc training from MIT. He had claimed that he had created the first human genetically edited babies. There are two twin baby girls. The baby's father was infected by HIV. Therefore, the initial thought was to create an offspring who has dysfunctional CCR5, similar to the CCR5 delta 32. And then in this case, they will never be infected by HIV. As I remembered, it turned out one baby had a heterozygous CCR5 deletion, and the other baby has a homozygous deletion. Okay, so this was a shocking news in the scientific field by then, and brought up a lot of ethic issues. Okay, following the inspection on how the experiment was executed, the He received widespread condemnation, and then he was sentenced to 3 years imprisonment, and also RMB 3 million yuan. That's about $500,000 US of fine. Okay, so this graph shows how just theoretically an embryo could be edited using the powerful tool CRISPR-Cas9. Okay, the CRISPR-Cas9 was delivered into fertilized egg to basically snip out the CCR5 DNA before the embryo was even divided into 4 individual cells. And it was hoping that this deletion of CCR5 can prevent HIV from entering the CD4 T cells. You can immediately see several problems with this case, right? So one is the function of CCR5 has not been completely studied. We do not know the function of CCR5 in cells other than CD4 T cells. And depletion of CCR5 from a zygote eliminates the protein from all cells in this person, which is potentially dangerous, right? The second reason will be CRISPR-Cas9 has off targets. Remember that CRISPR-Cas9 recognizes target sequences through the guide RNA. This is also called a gRNA, which has a complementary sequence with the targeting sequence, right? So if another location on the genome contains highly similar sequences, it may be edited too. So today, actually early this morning, I found that in this upcoming issue of Cell, there will be an article showing that the CRISPR-Cas9 could lead to frequently loss of chromosome when performing the human germline gene editing. This article will be published in the December issue of the Cell, but its online version is available now. So in this study, the investigators performed the CRISPR-Cas9 editing of this gene called EYS. And they editing this EYS gene to produce a frame-shift mutation in this human fertilized egg, similar to this He Jiankui did before. So we learned that CRISPR-Cas9 produces double-strand breaks, right? So it's double-strand breaks. Their results show that only approximately 50% of the chance the double-strand breaks failed. The double-strand breaks are repaired as intended, but in another 50% of the chance, the double-strand breaks there are failed to be repaired, and the unrepaired double-strand breaks persist throughout the mitosis and will result in frequent chromosome loss. Sometimes you lost like half of them, and sometimes the whole chromosome has been lost, okay? Therefore, they conclude that human germline gene editing would pose a substantial risk of aneuploidy, we learned that, right? And other adverse genetic consequences. At least currently, the CRISPR-Cas9 technology is not perfect, particularly for human gene editing. Also, a variation of gene therapy is enhancement gene therapy, meaning this gene is intact in the body, but the purpose of gene therapy is to enhance the traits. Okay, so this slide shows gene doping by the use of this EPO that is also called erythropoietin. So this is a hormone that increases red blood cell production to enhance the athletic performance, okay? Ripooxygen was the trade name for a type of gene therapy to produce this EPO, okay? Ripooxygen was originally developed for the treatment of anemia, but was never tested on humans. This product has been banned in 2003. There was controversial over using the gene therapy to transfer the gene for EPO, which would be undetectable if used, right? So it is not fair if any athlete is using this for, the competing games. But then, although germline gene therapy has many ethical issues, the combination of somatic stem cells and gene therapy has already produced its first commercial progeny, okay? In the 2016, a drug called Streamvilis was proved in Europe for the treatment of a condition called this adenosine deaminase severe combined immunodeficiency, or this ADA SCID. Okay, that's actually a form of disease initially afflicted David Vetter, the bubble boy we talked about previously. So during the clinical trials, all 18 children with this ADA SCID treated with Streamvilis between 2000 and 2011, they survived, okay? But in contrast to that, untreated babies with the disease typically die before age of 2, okay? So this picture shows you how the therapy works. So first of all, the stem cells were extracted from the patient's bone marrow. And then in the lab, a virus carrying the normal gene is delivered to the stem cells and inserted into their genome. So then the modified stem cells are put back into the baby's bone marrow, where they started to producing functional immune cells. If David Vetter was born like 3 years later, he would have survived. I will quickly go through the slides of genetic counseling. Okay, first, what information can genetic counseling provide? The genetic counselors can help people understand the occurrence and the risks of having a genetic disorder in their family. They can explain to the patients the medical facts, what diagnosis and treatments are available, how to deal with the disease, and the risk of recurrence. Okay. Now, if you learn this course well, you should be able to find all this information. And with some training, you should be able to work as a genetic counselor. During a genetic counseling session, the counselor uses the information from pedigree construction, medical records, and genetic testing to educate and inform a couple about their risks for genetic disorders. We learn all of this information in this class, right? So this slide lists who should seek for genetic counseling. I will not go through these one by one. The basic idea is that any risk that could bring in genetic alterations in the offspring can or should go to see the genetic counseling. Okay, now let's spend some time discussing an exciting field that receives a lot of attention in recent years. That is cancer immunotherapy. Okay, the development of cancer treatment has gone through several stages. As of today, cancer immunotherapy is a current and future therapy to treat cancer. So our textbook is outdated on this topic, and you need to review my slides or videos for this information, okay? So cancer immunotherapy is a therapy used to treat cancer patients that involves or uses components of the immune system. It aims to enhance the power of of the host immune system for the treatment of malignancy. So basically, there are two ways to make this happen. One is stimulating the body's own immune system to work strongly to attack on the cancer cells. Okay. The second is giving immune system components such as recombinant immunoglobulin proteins. Those are the two strategies. Cancer immunotherapy actually has a long history. As early as 2600 BC, Egyptian physician recommended producing a localized infection to promote regression of tumors. Then in the 1890s, a New Yorker bone surgeon named William Coley, who now known as the father of immunotherapy, started to treat his sarcoma patients with an immune stimulating bacterial mixture that came to be called Coley's toxin. Okay, Coley treated nearly 900 patients with the toxins over a 4-decade period, achieving a cure rate of more than 10%. That's actually pretty high at that time. This slide shows a simplified key events in the immuno-oncology timeline, from Coley's early discoveries through to some of the recent approvals for melanoma and lung cancer. Okay, showing here for the last 3 agents showing here. So the ipilimumab is an anti-CTLA-4 checkpoint inhibitor and has been used for melanoma treatment. Okay, and these two, this is nivolumab and pembrolizumab, are the anti-PD-1 checkpoint inhibitors and have been approved for treating melanoma and lung cancer. Let's now watch a short video to see what immune checkpoint means and how these inhibitors work.
One of the most exciting approaches to cancer immunotherapy involves releasing the natural brakes that keep the immune system in check. This approach, called checkpoint blockade, takes advantage of the fact that the immune system already knows how to fight cancer. It just needs a little help. The immune system's main cancer fighters are called T cells. T cells have receptors on their surface that do different things. Some act like a gas pedal on a car, telling the T cell to go. Others act like a brake, telling the cell to stop. Drugs called checkpoint inhibitors release the brakes, allowing the immune system to step on the gas and fight the cancer. Many cancers have learned to hijack these same immune brakes to avoid destruction. They make a molecule that engages the brakes. Checkpoint inhibitors block this interaction as well, robbing cancer of an important defense. Several checkpoint inhibitors are currently FDA approved for the treatment of melanoma and lung cancer. The drug Yervoy blocks a checkpoint called CTLA-4. 2 other drugs, Opdivo and Keytruda, block a checkpoint called PD-1. Many cancers respond to checkpoint inhibitors, and the future looks very bright for this important class of immunotherapies. Created by the Cancer Research Institute, the leader in immunotherapy.
So we learned about immunology last time, and these slides should be easy to understand. So based on the video, CTLA4 and PD-1 are both immune checkpoint proteins and both inhibit the T cell activity. But are they different or the same? The answer is different, okay? You can see from this slide, these two molecules function at different stages of T cell activation, okay? So CTLA4 on T cell interacts with the B7 molecule expressed on these antigen-presenting cells during the priming stage. And when T cells are initially activated by the pathogen peptides. But in contrast, PD-1 interacts with PD-L1 on targeting cell, which is a cancer cell in this case. So when the T cell is ready to kill the cancer cell. So therefore, the inhibitor drugs can release the brakes on T cells at different stages, okay? James Allison and Tasuku Honjo were awarded to the 2018 Nobel Prize in Physiology or Medicine for the development of the revolution in cancer therapy, the immune checkpoint blockade. Dr. James Allison is the chair of Department of Immunology at the MD Anderson Cancer Center, and he works on CTLA4. And Dr. Honjo is a professor of medicine in Kyoto University, and he works on PD-1. So this is basically a side note. This picture shows Dr. James Allison's car taken next to the TMC Transit Center, this picture. Okay, look at the plate. And immediately you recognize this is Dr. Allison's car. Okay, since we mentioned about car, now let's take a look at another new technology related to immunotherapy. So it is called a CAR T cell, okay? So this CAR means chimeric antigen receptor. So it was considered as the 2018 Advance of the Year by the ASCO, so the American Society of Clinical Oncology, that's the ASCO mean. So this picture shows a CAR T cell, which is yellow here, attacking a cancer cell. From last lecture, we have learned that T cells recognize the MHC peptide complex through this structure called a TCR, T cell receptor. And the interaction mediates the killing to target cells, such as cancer cells in this case. Therefore, the scientists thought whether we could engineer an artificial receptor that is similar to the TCR and specifically target known cancer antigens, resulting in more efficient killing of cancer cells. The answer is yes. So let's watch two short videos in a row. Okay, so the first one focuses on how the CAR T cell therapy is performed. And the second one shows how T cell recognize and kill infected target cells. So the killing is exactly the same for the CAR T cells to kill cancer cells. Okay, let's watch together.
The immune system defends the body against disease. T cells are one of its key soldiers, targeting infected or abnormal cells. But cancer can block those defenses. Now scientists are genetically engineering patients' own T cells to make them smarter and tougher at seeking out and destroying cancer. One version is called CAR T cell therapy. T cells customized to zero in on a patient's specific kind of cancer. It all starts when a patient's blood is filtered through a machine that separates T cells and other white blood cells from the rest. Scientists mix the collected T cells with a virus that has been disabled so it won't cause illness. Instead, that virus carries genetic instructions for the T cells to grow an artificial receptor called a chimeric antigen receptor, or CAR, that will track its cancer target and rev up for an attack. Millions of copies of those engineered cells are grown in a laboratory and given back to the patient intravenously. The first CAR T cells are being tested against types of leukemia and lymphoma that bear a marker or antigen named CD19. Once in the bloodstream, those CAR T cells home in on that antigen and grab hold. They release toxic chemicals that trigger a cancer cell's death. As one cancer cell dies, CAR T cells move on to the next, multiplying in the patient's blood until they're no longer needed.
So this video shows how cytotoxic T cells function to kill the target cell. So this blue cell is a cytotoxic T cell. And then when it sees a healthy cell, the TCR will not recognize the self-antigen presenting in the MHC1 molecule. However, when it sees a virus-infected cell, which is presenting the viral peptide on the surface, it will recognize this antigen, and then this interaction will activate the cytotoxic T cell and secrete a lot of cytokines. And then the effector function, including the secretion of two enzymes, perforin and granzyme. So the perforin actually drill holes in the surface of the target cells, and then the granzyme will enter into the target cell and lyse the target cell. Then here comes a question. How CAR is designed? CARs combine many facets of a normal T cell activation into a single protein. It is composed of 4 regions. First, this is an antigen recognition domain that binds to target cell surface antigen. Second is an extracellular hinge region that provides flexibility so that allow the first antigen binding domain can have flexibility, easier to target the antigen, okay? And then the third is a transmembrane domain that localizes the molecule on the cell membrane. And then lastly is this this is intracellular T cell signaling domain that mediate the T cell activation. Okay, for the extracellular antigen recognition domain, scientists took advantage of antibodies. We learned antibodies, right? Because antibody can bind to antigens through these variable regions, also named the FAB domain. So that antibody can bind antigen very specifically, okay? So the 2 subunits of variable regions can be linked together artificially with this linker sequence, okay? So they therefore become a molecule called SCFV. Also, that means the single chain fragment variables. So therefore, the SCFV domain has this ability to recognize and bind to targeted molecule or the antigen very specifically, okay? So once the SCFV domain brings the CAR T cells to its target, how can the T cells be activated and kill the targets? The CAR T cells need the signals to be activated so they can kill those cancer cells. This is similar to the TCR activation, and this process needs 2 signals. The signal 1 activation is mediated by these CD3 zeta subunits in the whole TCR molecule, okay? And then the CD3 zeta signaling is the major signaling for T cell activation. Therefore, scientists were engineered this protein CD3 zeta into the intracellular components in the CAR. And then in order to have the maximum activation of T cell, so it also needs signal 2, also called the signal 2 co-stimulation. So this is mediated by the co-stimulatory factors such as the 4-1BB or CD28 molecules. So the scientists then using genetic engineering of those two molecules together into this CARP, and then combine all the molecules together. In this case, the CAR molecule will have both signaling 1 and signaling 2 stimulation. So it can be fully activated once this SCFV bind to antigen, and then this will activate the CAR T cell, therefore to kill the cancer cell, right? So this slide shows several generations of CARs. First generation of CAR contains solely the CD3 zeta chain as the single activation domain, leading to T cell activation and cytotoxicity based on the SCFV specificity. All right, and the second and the third generation of CARs consist of either 1 or 2 additional co-stimulatory signaling domains, respectively. So such as the CD28 shown here, or OXY40, also OXY40, another name is the CD134, and also 4-1BB as we shown in the last slide, okay? 4-1BB has another name called the CD137, okay? So, but this, you don't have to remember this CD numbers, you just remember there is signaling 1 and signaling 2, and you know, the second signaling 2 actually can enhance the overall the survival as well as proliferation and persistence of activated T cell. Okay, so showing on the bottom. So the function, remember the function. Remember that we have several generations of CARs. Okay. So currently, all the CARs available or tested by scientists they're trying to develop is the generation, second generation, and the third generation. So many scientists have contributed to the CAR T technology, but I would like to introduce a few of them. So the first is Dr. Kajun. So he obtained his medical degree from Baylor College of Medicine, and currently a professor at UPenn. So in the 1980s, his lab discovered the CD28 molecule as the major co-stimulatory signal for T cell activation. A few years later, he tested the ability to culture genetically modified CAR Ts in humans, discovering the cells could engraft and persist in patients with HIV or AIDS for years. Okay, his work led to the development and the commercialization of the Kymriah, so the first FDA-approved gene therapy. So we will talk about this drug in a couple of slides. And then Dr. Michael Sutherland at Memorial Sloan Cancer Center was the first to report the design of second generation of CARs that has a co-stimulatory domain designed to promote the cell proliferation and the survival. Okay, and then Dr. Dario Campana from the National Institute of Singapore made the first anti-CD19, 4-1BB and CD3 zeta CAR that was highly effective in treating patients with an aggressive form of leukemia. In the medical history, there always has some patients that were unlucky to develop deadly diseases, but they are luckily cured with new treatments. So here comes two cases. The first one, the Bill Ludwig, was diagnosed with chronic lymphocytic leukemia, also called CLL, in 2000. The CLL is a cancer of B cells, okay? Bill then went through rounds of chemotherapy every few months when his B cell count grew too high. The cycle took its toll. So in 2010, Bill was enrolled in the first clinical trial of CAR-T directed by Dr. Jun, the CAR Jun we just introduced. And then Bill was cured, and so far so good. The other case is Emily Whitehead, who developed acute lymphoblastic leukemia, also called ALL, in 2010. Okay, in spring of 2012, her cancer relapsed twice after chemotherapy. So Emily's parents ran out of choice and sought the CAR-T therapy, which had never been tried in a child or in anyone with this type of leukemia. But the treatment almost took Emily's life. Here is a small story, okay, share with you. So a sign that the treatment is working is that the patient becomes terribly ill with raging fevers and chills. So its medical name is called cytokine release syndrome, or you sometimes heard it's a cytokine storm. So referring to the cytokines releasing from immune cells as they are activated, causing fevers and other symptoms. The storm can also flood the lungs and cause dangerous drops in blood pressure. So effects that nearly killed Emily. Steroids sometimes can ease the reaction, but they did not help Emily at that time. So her temperature hits 105 degrees. So her friends and family who had come to say goodbye to Emily. But blood tests gave the researchers a clue as to what might help save Emily. Her level of one of the cytokines named interleukin-6 or IL-6 had shoot up 1,000-fold, okay? So doctors had never seen such a spike before and thought it might be what was making her so sick, okay? Importantly, Dr. CAR Jun knew that the drug could lower the IL-6 called tocilizumab. So his daughter took it for rheumatoid arthritis. So they ordered the drug and used it on Emily. The response was amazing. So within hours, Emily began to stabilize and she wake up a few days later. So far, her cancer never comes back. Every year after that, Emily took a picture to show the milestones she made through. So this picture, 8 years after this cancer-free, that's Emily took on September this year, in September this year. So since Emily's story, tocilizumab has been routinely used in CAR-T therapy to treat the cytokine storms. The story also tells us the importance of being a good doctor. Your decision could determine a person's life, sometimes even an entire scientific field. So up to today, FDA has approved 2 drugs for CAR-T therapy. We saw the video actually talk about this. So one is Kymriah, approved in 2017. And this was the first drug that cured Emily's leukemia. So this drug can achieve 80% of remission in ALL, but a little less efficient on diffusive large B-cell lymphoma, also called a DLBCL, in which 1/3 of a complete response and 18% of partial response is achieved. However, another approved therapy, Yescarta, has better performance in DLBCL treatment. You can see 51% of patients show complete response and 21% of patients show partial response in clinical trials. Both CAR targets a B-cell specific surface marker, that is CD19, okay? Because both cancers are B-cell cancers and all cancer cells express CD19, making CD19, this molecule, a classic biomarker for immunotherapies. All right. So although T-cell therapy is very effective, it is also very costly. So Yescarta costs about $373,000 to treat adults with advanced lymphomas, while Kymriah costs $475,000 to treat acute lymphoblastic leukemia in children and young adults. In addition, many patients experience serious side effects that can land them in hospital intensive care units for weeks, pushing treatments cost it's about more than $1 million. So this is the last slide that shows the efficacy of Yescarta on a DLBCL patient. The patient went through several rounds of chemotherapy and radiation therapy before the CAR T therapy. You can see that 3 months after the Yescarta treatment, actually most of the cancer cells were gone, okay? He remains in complete remission 9 months after Yescarta treatment, this patient. All right, that's all for today. Hope you enjoy it.